patch clamp technique. This section comes under Unit 13 for CSIR net, and the section name is Methods in Biology. So let's quickly look at the syllabus. So this is the G part of Unit 13, and in this particular part, we would learn electrophysiological methods such as single neuron recording, patch clamp recording, etc. So this video is focused on this topic. Now there are not many resources that has very good explanation of these techniques. So in this video, you are expected to learn that. Let's quickly browse through the analytics. It's in the Unit 13. Questions asked in part C, many of the cases, and also in part B. Generally, the questions are towards the difficult side, but sometimes they also ask easy questions. Now, it would be easy if you learn the concept from this video. And obviously, application-oriented questions are generally asked, and there could be also principal or technical questions as well. So let's learn the concept in 15 minutes. We'll learn what is patch clamp, working principle of patch clamp, application of patch clamp, and limitations of this technique. Patch clamp is an electrophysiological technique which allow us to understand what are the physical properties of the neuron. So let's quickly review the patch clamp technique. The patch clamp technique allows the investigation of small set of ion channels, even sometimes single ion channels, and researchers can try to understand the kinetics of ion flow through this channel. Overall, it can give us an idea about the excitable properties of a cell, not only neurons but any other cells such as cardiomyocyte, muscle fibers, etc. Now, patch clamp experiments are performed from cultured cells, from tissues, from tissue slices, and dissociated cell cultures as well. So let's quickly look at the applications before we go into details. So first of all, we can investigate ion channels of excitable and non-excitable cells using patch clamp method. This is a very important method for medical research because many diseases are associated with the malformation of or malfunctioning of the ion channels. So in order to understand the ionic properties, this particular technique could be useful. In pharmaceutical research, it is very important and interesting because many drugs operate by targeting several ion channels. So learning the basics of how ion channels work, what is their electrophysiological properties, is important to design those kind of drugs. Lastly, it is really important for neuroscientists because if they want to know a property of the neuron, property of the circuit, what are the neuronal changes underlies a behavior, then this kind of technique combined with calcium imaging is super important. Quickly describing the resources that you can possibly use. You can use the conventional Pathfinder book set for the CSIR net. Among these, you can look at the biophysics and molecular biology book for a detailed overview in this topic. Anyway, the Wilson and Walker book can also be useful, but again, none of these books discuss this topic in details. These biophysics, molecular biology books describe this topic in short. You can find that in chapter 9, page number 129. But these topics are described in details in Neuroscience 5th edition, PERVs, or in Eric Kendall's book. So if you want to learn this topic in bit more details, you can refer to these books all the time. Every link is provided in the description. Question is, can we measure action potentials using patch clamp? The simple answer is yes, we can. Not only action potential, many other electrophysiological properties such as capacitance, conductance, firing frequency, input resistance, firing pattern, all of these can be understood from the patch clamp method or the variations of patch clamp method. So we would learn about all these properties sequentially. So stay tuned because these are not really discussed in the books. So let's try to understand the patch clamp setup to prime ourselves towards the technique. So this is how a ideal patch clamp setup look like. You can see the microscope. You need to look at the neurons or patch the neuron under the microscope. So obviously you need a very good microscope. And this microscope and the entire setup is kept under a Faraday cage. Now it is mounted on a anti-vibration table, because while finding or patching a neuron, minute vibration matters. And all of these setup is connected with an amplifier setup, because first of all your signals are so low, you first need to amplify the signal in order to visualize something in the computer, and it is attached to a software which is controlling these amplifiers and other electronic components. Then there is a sample preparation table where you prepare the samples, you bevel your electrodes, all of these things are there. And inside the Faraday cage, you can see on the left hand side there is a perfusion setup. That means you can run several solutions through your chamber. For example, KCl, NaCl, or several salt solution, toxins, other pharmacological drugs, agents, etc. Now let us zoom into the objective and the particular patch clamp recording setup. Now this is important to understand because this entire setup is mounted on a customized microscope stage. You can see the sample, in this case a mouse brain tissue, a mouse brain vibrotome section, is now embedded in an artificial cerebrospinal fluid. And the reason behind putting it in an artificial cerebrospinal fluid is to keep it alive. If the section or the neurons are not alive, how we are going to determine the electrophysiological property? They need to be alive. Then you can see a micromanipulator. Micromanipulator is a device which allows the pipette and the electrode to move in a precise fashion. And you can see using the micromanipulator, one can adjust the pipette and adjust the electrode. And everything is mounted under a microscope objective. So one side we can visualize and one side we can try to patch or find the cells using these electrode. So recording action potential is completely possible using patch clamp setup. You can record action potential, firing pattern over time. And in order to do that, you patch a small portion of the neuronal membrane. So here is the electrode, and here you can see a small patch of the neuronal membrane which is electrically isolated. Now suction is applied when the electrode approaches the neuron, and this creates a very high resistance seal around the sucked membrane. And this resistance is so high, it is in order of gigaohm. Now this tight seal is really important. This tight seal ensures that this portion of the membrane is electrically isolated, and all the ion that moves through this particular region moves into the electrode and nothing leaks out. So moral of the story, in order to first establish a patch, you have to ensure nothing leaks out, and the patch is the particular gigaohm seal is this gigaohm seal is nice and it is not leaky. It has to be established properly. Now let's talk about the technical details of patch clamp. So how patch is established, that is really important. So in order to establish a patch, you start with a particular electrode. You try to find the neuron under the microscope stage. You put a small voltage, you see a current like this in blue. Now here, next, when you reach the neuron, then you apply a voltage. In this case, sorry for the mislabeling, the I and V got just opposite. So I consider I to be V and V to be I. So anyway, look at this. When you approach the particular neuron, you apply another voltage pulse, but you get a small current pulse. Why? Because there is a resistance due to the neuronal membrane, and that is why V equal to IR, right? So if the resistance is high, obviously the current has to be low. That is why we see a low current. Here the current trace is in red. Throughout this diagram, the current trace is in red. And then when you apply a brief suction, the membrane breaks open and you can see transients of current that starts flowing. This red particular trace shows a transient current. Again, the labeling is not correct, so that's why don't get confused. Okay, so the first thing that we learned that we have to patch a portion of the membrane using this electrode, which is pretty difficult to locate under a microscope. Then the electrode is the main part. In the electrode, there is specific solution that we load the electrode with. This is called pipette solution. And the entire setup is now attached with a feedback resistor, overall amplifier setup. Sometimes there are also feedback resistors to achieve specific kind of task. We'll go to that in details. But anyway, the amplifier is attached because the signal coming out of these pipette is very low. And these kind of electronic signals can only be detected if you amplify it with the amplifier circuit. Now the glass pipette contains electrolyte solutions, and this particular pipette ensures that it isolates a portion of the membrane and make it electrically isolated from the other regions of the neuronal membrane. Now currents fluxing through these ion channels flow through the pipette and gets recorded and amplified, and that's what we see as a signal. Now there are different subtypes of this patch clamp. So overall patch clamp setup and technicality is now clear. We also looked at how to establish a patch, but there are different operation regimes of this patch clamp method, such as cell attached patch clamp. In the cell attached patch clamp, you just attach the pipette with the neuronal membrane and create that gigaohm seal, and that's what it is. So whatever flows through the ion channels in that portion flows through the pipette, and you record that. That is called cell attached recording setup. The second one is whole cell patch clamp. In this case, you start like a cell attached recording pattern. You establish the gigaohm seal, but then you further apply a brief suction. Brief intense suction breaks the membrane and makes the neuronal interior to be continuous with the pipette solution. So overall neuronal property can be determined using a whole cell patch clamp. Then there are two operation regimes known as inside out and outside out patch, and we would look at their utility in bit more details. So in inside out patch, what is happening is like a portion of the membrane, the inside portion of the membrane, look at the cytoplasmic side of the membrane, that is exposed. Okay, and in the outside out patch, the outside portion of the membrane, which was there, which was pointed towards the outside in an intact neuron, is now outside. Okay, so let's talk about the cell attached patch clamp in bit more details. So when the pipette is in the closest proximity of the cell membrane, mild suction can lead to tight seal. We'll call it the gigaohm seal, and that is a cell attached recording setup. In the whole cell, we start like a cell attached, but apply brief suction which breaks the membrane, makes it continuous with the pipette solution. Now whole cell patch clamp recording is most frequently used recording setup. Now cell attached patch clamps are used to observe the gating of ion channels. When it is unknown, the gating of the ion channels can be probed using a cell attached patch clamp. Whereas the whole cell patch clamp gives us an overall idea about neuronal property. In a moment, we would expand and elaborate on whole cell patch clamp because that's the most important in context of CSIR net. There is also inside out and outside out patch clamp. So in the inside out patch clamp is very useful to look at some kind of like, since the inside portion of the membrane is out, then you can dip this particular thing into a solution containing many different things. So it is used to understand what chemicals can modify ion channel activity intracellularly. And outside out patch, the outward portion of the membrane patch is exposed. So you can again dip it into a solution that might contain some neurotransmitter, some peptides. So overall, how neurotransmitter interacts with the channel and how it changes the channel kinetics. If you want to study those kind of studies, then outside out patch clamp is super important. So you can understand that outside out and inside out, these kind of specific settings are really useful for understanding channel kinetics. How the channel is modulated from the inside of the cell or the outside of the cell. You can use you can use these techniques to understand and study those questions. Now, the most frequently used technique under patch clamp is the whole cell configuration. So we call it a configuration because in whole cell configuration, we are still doing the patch clamp, but one small operational changes. Now here the membrane patch would be broken down with the application of breath suction, as you can see here. Now there are two operational regime under whole cell configuration. There are two modes, I would say. So first is voltage clamp mode, another is the current clamp mode. So whenever we say voltage clamp or current clamp, that means that particular parameter is constant. For example, in voltage clamp, voltage is constant. In current clamp, current is constant. Now in the voltage clamp method, we give a brief, we give a stimulus. So obviously we should record the current, and here the current is measured as picoampere. It's very easy to read this graph. So here you see the scale bar. That means that portion of the scale determines time in terms of minute. So if you take this scale and place on this trace, you can see this particular trace is roughly about 30 millisecond. And the amplitude is determined by picoamps of current. So if you again place this particular stick into the curve, you would see, okay, it's roughly around like 180 picoamp is the peak amplitude, roughly. So now you look at the current clamp mode, your current would be constant, you are measuring voltage. So voltage fluctuation is nothing but the action potential. So see, you give a stimulus and there is voltage fluctuation, and that is the action potential you are recording. And again, the axis is like millivolts millisecond. So now you can see how the current clamp setting is working. You hold it into different voltages and you are measuring currents. And in certain particular, you are holding it into different current and measuring voltage. So the lower curve, you can see the output is the voltage output, and you see the action potential beyond a particular level, right? So you see how current is changing. How the voltage is changing when we keep the current at a particular stage. And that can be understood from this particular voltage versus current curve, and how we can see how the curve is changing. Okay, now let's talk about examples of patch clamp recording. Let us take some live biological examples. So this is a glutamatergic synapse where there would be several ionotropic glutamatergic receptors. Which are just ligand-gated ion channel like AMPA receptors. So they bind to glutamate, they open up, and the cations like sodium, potassium can flow in, and this can depolarize the postsynapse. Now there are also NMDA receptors, which are also ligand-gated ion channel. They bind to glutamate, but they doesn't open so easily because there is a magnesium block present there. So initially nothing can flow through the NMDA receptor, but when there is enough amount of positive ions diffused through this AMPA receptor, the inside become positive. Now magnesium ion gets electronically repelled by these positive ion inside. Then it allows specific cations to get in. Now let's talk about the electrophysiological properties of AMPA receptors. So in AMPA receptors, this is what? A current clamp or a voltage clamp? You should be able to tell me right now by looking at the axis. Yes, this is a voltage clamp. We hold the voltage and record the current. So you can see we hold the voltage at minus 70 millivolts. We record a current. We hold it at minus 50. We record another type of current, but see the amplitude is changing. And this is how we just change the particular voltage input that we are giving. And we are getting different current input. We also see the directionality of the current is changing. So these are raw traces of electrophysiological traces would look like. This is how the raw electrophysiological traces would look like. Now you can get a better understanding of this property by plotting these kind of graphs known as IV plot or GV plot. IV plot is nothing but plotting the data. So you can plot I by checking the amplitude from this curve. So you see the maximum dip or the maximum up, these kind of regions are the amplitude of this graph. So you can just plot it in the y-axis and plot the voltage. So you know the voltage because you are holding the voltage. So you plot 70, minus 70, you plot a amplitude of the current, and then gradually after plotting it, you would see a curve like this. So these are the IV characteristic curve. So this is one electrophysiological signature of glutamatergic neuron. Anybody would see this particular curve and they would say, oh, this is a AMPA current. Anyway, you see these AMPA currents are not dependent on voltage. So the conductance through this channel is not at all dependent on voltage. It's a flat line. You change the voltage, nothing happens to the conductance. So it's expectable, right? Why? Because it's a ligand gated ion channel. It's not a voltage gated ion channel. That is why even if you change voltage, nothing happens to the conductance of this channel. I hope you understood this detailed property in bit more. So here we saw the inward current in negative voltages, whereas outward current in positive voltages. So these kind of information can be extracted using these IV plot or patch clamp recording. Now you can also look at the NMDA receptor kinetics. NMDA receptor kinetics somewhat looks like this. Even if you don't know anything about the NMDA receptor properties, from these electrophysiological graphs you can make out that almost no current is present at a negative voltage. Now when the voltage becomes a bit more positive compared to minus 100, for example in a range of minus 50 to 0, which is above the membrane threshold, resting membrane potential, or reaching the action potential threshold, there you can see small amount of inward current is flowing through it. But the current is high in amplitude and it is outward in direction when the voltage is positive. So this tells you that NMDA receptor only operates when the inside of the membrane is pretty much positive. And that's what we see in that cartoon. Remember? In order to NMDA receptor for function, there should be enough amount of positive charge build up previously by AMPA receptors. So these graphs tells us how NMDA receptor works. And also the conductance versus voltage plot tells us there is almost no conductance when there is negative voltage. So now we can compare these kind of graphs. For example, we can compare the AMPA and NMDA. We can compare their overall IV characteristics, GV characteristics. So this is something very known. Think about some unknown channel you are working with. So you have to plot these kind of graphs to understand or to describe their electrophysiological properties. So I hope this part is really clear. So why one should really bother to learn about these things? And let me explain how. So let's say you are doing a wholesale patch clamp recording and you want to isolate the AMPA current from the NMDA current. You only want to record the AMPA current. So obviously if you do it on plus 60 millivolt, you get a current trace like this. But how do you separate the current? Like, how do you know this particular current that I'm seeing in my electrophysiological recording, is it coming due to the AMPA receptors or due to the NMDA receptors? So there is a very easy trick that the electrophysiologists do. In order to look at the AMPA current exclusively, they record at minus 60 millivolts, because from the IV curve they can see that at negative potentials there is no current through the NMDA receptor. So obviously the current would be exclusively through the AMPA receptors. Now once they know the AMPA receptor current, that means I minus 60 millivolt is actually AMPA current, and I plus 60 millivolt is a mixture of AMPA and NMDA current. So just by subtracting I plus 60 minus I minus 60 millivolt, it would give you NMDA current. So AMPA and NMDA current can be simply calculated using these kind of tricks. So this is what is used in the field of electrophysiology. So now you can understand there are several other summaries that they would look at. They can look at the conductance of these channels. Anyway, the conductance is like sodium and potassium for AMPA receptors, and for NMDA receptors it is sodium, potassium and calcium. These graphs also give you the reversal potential, but anyway, these graphs doesn't tell you about what ions are conduct. I'm just giving this information to you. Single channel conductance can be recorded using patch clamp technique. Also, we looked at the current kinetics. So in case of AMPA, this is a fast opening, fast closing channel. So the current starts and finishes very quickly. But if you look at the NMDA kinetics, the current starts, or the channel opens very quickly but closes very slowly. So it's a sustained current. Again, these kind of properties can be understood from these kind of patch clamp recording setup. So I hope this was useful in terms of understanding the concepts. Let's look at some previous year questions. A neurophysiologist was interested in using patch clamp technique. Following statements are related to the technique, and these statements are: intracellular movement of ion through channels, post-translational modification of ion channel proteins, ligand that controls the opening and closing of the ion channel, change in current flow in a single ion channel. Which of the following combination will be achieved using patch clamp method? So the correct answer will be option C, R and S. So ligand that controls opening and closing of ion channel, we can simply understand from this video. We can use those configurations, inside out and outside out, right? So specifically ligand, if you want to understand, outside out would be the best method, because the outside of the channel is now outside of the pipette. You can dip it into the solution containing several neurotransmitter, and that is how you can understand how a ligand modulates an ion channel. And S is important because change in the current flow in a single ion channel can be recorded using patch clamp. We looked at it because all these inside out, outside out recording setups does this. So this was asked in June 19, part C. Though it is asked in part C, it's not that difficult, right? Okay, let's look at some of these questions which are CSIR pattern. So yes, part B style question. Which of the following is not an application of patch clamp technique? So application oriented stuff is really important. A, recording the action potential of the neuron. B, recording single channel conductance, investigating neuronal morphology changes, investigating the influence of extracellular cues such as neurotransmitters. If you understood the technique correctly, you should be able to pick the answer C. Morphological properties are not analyzed using patch clamp. The electrophysiological properties are analyzed. Question number 2. Identify the mismatched pair. Inside out patch clamp, measure single channel activity. Outside out patch clamp, investigating the influence of extracellular cues such as a neurotransmitter. Wholesale patch clamp, the interior of the pipette becomes continuous with the cytoplasm. Cell attached patch clamp, the cytosolic surface of the membrane is exposed. So if you learn properly these modalities of patch clamp, then you should be able to answer. The correct answer here would be option D. And this is a part C style question. Let's see whether you can answer it or not. I would leave it on you guys. So given are the traces from a neuronal culture after treatment with a drug X. Recording is done in whole cell configuration. And it is also mentioned that it's a current clamp mode. What can be concluded from these traces? You can see the traces, vehicle, that means the control thing is basically in green, and the drug treated one is basically in red. So what we conclude from these traces? The options are: firing frequency has decreased upon treatment with drug X. Input resistance has decreased upon treatment with drug X. There is no change in membrane conductance, and all of the above. So if you understood the video correctly, you should be able to answer this question, and I'm leaving it on to you. Put it in the comment. Anyway, this is a quick summary for patch clamp technique that we have learned so far. I hope this was a comprehensive summary. You can get many notes and flashcards in my Facebook channel. You can get that particular summary statement in the Facebook channel as well. You can follow me on Instagram, Facebook. All the links are in descriptions. You can support the channel by paying via super thanks option, which is present in the bottom right corner of any video. You can pay via Paytm, PayPal, UPI, net banking, etc. So you can support me in Patreon. All my social media links and the Nerdmedic channel's social media links are provided in the description. If you wish to connect, you can connect anytime. Feel free to connect. So see you in the next video. But before leaving, let me tell you, making one of these videos requires a lot and lot of effort, lot of reading, lot of designing the presentations, creating these graphics. So each of these lectures takes about 2 to 3 days to make these. So please share it with your friends. Let me know in the comment whether it is useful or not. Anyway, see you in next video.